Hello and welcome you all to my channel Omni Bio Entrance, and today we will be discussing CUCET, that is Central University's Common Entrance Test. This is an MSc Entrance Test question paper. This is for the subject Biochemistry, conducted in the year 15th June 2023. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CUCET MSc entrance exam, then you can refer to my channel playlist. My channel name is Omni Bio Entrance, and in that you will find the channel playlist where you can find these question papers. Coming to the first question. In E. coli lac operon, the lac repressor bind to the operator region in the absence of lactose. So, in the absence of lactose, the lac repressor will bind to the operator region, preventing the binding of RNA polymerase and thereby preventing the transcription and translation of the lac genes. Now, when lactose is present, lactose will bind to the repressor and prevent the repressor from binding to the operator region. Now, in the absence of lactose, they have asked. In the absence of lactose, the lac repressor bind to the operator region very strongly because the repressor has got more affinity for the operator region. So the answer here is option 2. Very strongly. Then next one. One of the major drawbacks of transgenic plants or crops is they are often observed to be less productive. No. They are more prone for diseases. No. They may indulge in unwanted horizontal spread of transgene. That is correct. That is one of the major drawbacks of transgenic crops. They often produce poor quality seeds. No. Then next one. The protein synthesis process in prokaryotes is unique because in them. So in protein synthesis in prokaryotes, there is the transcription is coupled to translation. Whereas in eukaryotes, transcription take place in the nucleus. From there, the spliced mRNA is transported to the cytoplasm where it binds to the ribosomes and then get translated. So it is not coupled in the case of eukaryotes, whereas in prokaryotes the protein synthesis is coupled. So transcription is coupled with translation. Answer is option 3. Identify the biocatalyst among the given 4 examples. Lysosome, it is an organelle. Ribozyme, it is a biocatalyst, it is an RNA molecule. Chromosome, you all know. Golgi vesicle, again an organelle. In a Ramachandran plot, the angle of rotation lies between. So it lies between minus 180 degrees to plus 180 degrees. Answer is option 2. The following chemical compound is used in gene transfer using physical delivery methods. So for physical delivery techniques or methods of gene transfer, you use polyethylene glycol. Answer is option 2. Then next one. During cell division, protein degradation, proteolysis is facilitated by. So it is by the ubiquitin pathway. Option 1, that is amino acyl synthetase pathway, it is involved in protein synthesis. Restriction endonuclease pathway, it is related to DNA restriction and modification, not protein degradation. ATPase pathway, ATPase are enzyme that hydrolyzes ATP, but this is not directly involved in protein degradation. The answer here is ubiquitin pathway. Then in next one, in Sanger's DNA sequencing, for chain termination of replication, dash is used. It is the 2', 3' dideoxy analog. Answer is option 2. If you know that it is the dideoxy analog that is used, only one option is having the dideoxy analog in there. Whereas other options, dideoxy is not mentioned. So the answer also is option 2. Then next one. The commonly used antibiotic erythromycin is produced by. So erythromycin, it is produced by Streptomyces erythraeus. Answer is option 2. In nucleosome model, the double-stranded DNA is wound on which molecular structure to give organization to DNA and into chromatin. So in nucleosome model, the DNA is wound around histone octamer, whereas H1 is an linker molecule. So the answer here is histone protein octamer. Answer is option 2. Then assertion, reason, question. So assertion. The second law of thermodynamics states that total entropy of a system plus that of its surrounding always increases. Yes, that is correct. Reason. The release of water from non-polar surfaces responsible for the hydrophobic effect is favorable because water molecules free in solution are more disordered than they are when they are associated with non-polar surfaces. That is the correct reason for the assertion. So both A and R are correct, and R is the correct explanation of A. Then next one. Again assertion, reason, question. So assertion. Substrates are bound to enzyme by multiple weak attraction. Yes, they are not non-covalent interaction. They are non-covalent interactions. The non-covalent interaction in enzyme-substrate complex are much weaker than covalent bond which have energies between minus 210 and minus 460 kJ per mole. So assertion is correct. The mechanism of catalysis is dynamic, involving structural changes with multiple intermediates of both reactants and enzyme. That doesn't explain the non-covalent type of interaction between enzyme and substrate. But R is correct. So both A and R are correct, but R is not the correct explanation of A. Answer is option 2. Then next one, again assertion, reason. Nerve impulses are communicated across synapses by small diffuse molecule called neurotransmitters. Yes, there are acetylcholine is a neurotransmitter. So acetylcholine receptor is best understood ligand gated channel. Okay, that's also correct. Acetylcholine opens a single kind of cation channel called acetylcholine receptor, which is almost equally permeable to Na plus and K plus. That is correct. Now reason. P-type ATPases, which include the sarcoplasmic reticulum, Ca2 plus ATPase and Na plus K plus ATPase, are integral membrane proteins with conserved structures and catalytic mechanism. This statement, it doesn't explain acetylcholine receptor. It just gives a statement about the receptors. So both A and R are true, but R is not a correct explanation of A. Answer is option 2. Then next one, again assertion, reason, question. Assertion. Enzyme sucrase will not bind and act on other disaccharides such as maltose. That is correct. Sucrase is specific to the sucrose. Due to enzyme specific functionality, enzyme sucrase acts only on sucrose. That is the reason why it doesn't bind to maltose, because it is having enzyme specific functionality. So both A and R are true, and R is the correct explanation of A. Then next one, assertion. The number of adenine bases in DNA molecule is equal to thymine bases. This is as per the Chargaff rule. So as per Chargaff rule principle, in a given DNA molecule, the proportion of purines and pyrimidines is always equal. Adenine is a purine, thymine is a pyrimidine, both are equal. So as per Chargaff rule, it is correct, and that is the reason for the assertion. So both A and R are true and R is the correct explanation of A. Answer is option 1. Then next one, assertion. In yeast, a single CDK, that is CDK1, controls the cell cycle progression. That's correct. In mammalian cells, minimum 2 or in special situation more than 2 CDKs control cell progression. That is also correct. But they have not mentioned anything why only yeast is having a single CDK. They have mentioned about mammalian cells in the reason. So both A and R are true, but R is not the correct explanation of A. Answer is option 2. Then next one, assertion. Plant raised in vitro through tissue culture technique often exhibit morphological and genetic variation. That's correct because they show what is called as somaclonal variation. Plant genetic fidelity often gets impacted by somaclonal variation. That is the reason for the assertion. So both A and R are true and R is the correct explanation of A. Then next one. Arrange the following events occurring in the JAK-STAT signaling pathway in the correct sequence. So JAK-STAT signaling pathway is as given in this photograph. So you can see that when the receptor cytokine bind to the cytokine receptor, they get dimerized. The JAK will autophosphorylate its receptor. Then STAT will bind there. JAK will phosphorylate STAT. Then STAT will get dimerized. This phosphorylated STAT will get dimerized and it will be translocated to the nucleus where it will control transcription translation of the genes. So that is what is happening in JAK-STAT signaling pathway. Now we will see the correct sequence. So binding of ligand to the receptor, that is first one. B is the first one. So option 1 and 4 can be eliminated. We will see 2 and 3. B. After binding of the ligand to the receptor, there is dimerization of the receptor. C. So option 2 is the answer, not 3. Because 3 they have given phosphorylation of tyrosine residues. That will happen only after dimerization of the receptor. So answer is option C. Then only phosphorylation of tyrosine residues on the receptor by JAK. Then binding of STAT to phosphorylated tyrosine and phosphorylation of STAT by JAK. Then dimerization of phosphorylated STAT followed by translocation of STAT dimer to nucleus to modulate gene expression. Answer is option 2. Then next one. Protein destined to be secreted move through the secretory pathway in which following order. So which of the organelles are involved in transport of proteins to be destined to be secreted through the secretory pathway? So first will be the rough endoplasmic reticulum. So B starting itself is given only in option 4. So B should be your answer. So B will be first rough endoplasmic reticulum, then comes transport vesicle, then comes Golgi complex, then comes secretory vesicle, and then the protein is being transported to the cell surface. Answer is option 4. Then next one. Arrange the enzymes in sequence on the basis of their involvement in citric acid cycle. So this is the citric acid cycle. First is aconitase, then comes isocitrate dehydrogenase, then alpha ketoglutarate dehydrogenase, succinyl coenzyme A, then succinic dehydrogenase, then fumarase, malate dehydrogenase, citrate synthase. So first will be aconitase. So A. Then comes isocitrate dehydrogenase. You can see in the figure here isocitrate dehydrogenase B. Then comes succinate dehydrogenase E. Then comes fumarase. Then malate dehydrogenase. So the answer here is option 3. Then next one. Write the correct sequence of intermediates formed during pentose phosphate pathway. So pentose phosphate pathway, it is having a reductive phase and an oxidative phase. So first will be the oxidative phase and it will have glucose 6 phosphate. From there to 6 phosphogluconate. So D then A. So that is there in option 4. So first two itself if you identify you will get the answer as option 4 because option 4 is having D first. Then comes ribose 5 phosphate, then ribulose 5 phosphate and then pseudoheptulose 7 phosphate.